Hello and welcome to this undergraduate skills video where we're going to learn about various basic laboratory calculations which will be extremely useful as you progress through your degree. Now, why do you need to know about these calculations? Well, it will allow you to analyse scientific data, and once it has been analysed, you can then start to understand it, and once you understand it, you can start to draw conclusions on what the data might show. So without further ado, in this particular video, we're going to focus on calculating the variance, and for this you will need to understand what the arithmetic mean and normal distribution are. Therefore, I'd advise you watch the associated video before continuing. So before we take a look at variance, we need to understand what we mean by the term deviation. Now, on a very basic level, deviation is a term we use to describe the difference between an individual data value and the mean value of the dataset. And just to visualize this, here we can see two data points in yellow, and the deviation is the numerical difference between the data value and the mean value. So I guess a key question now is, how does this relate to variance? By definition, variance is a measure of how dispersed or spread out all of the data values are from the mean value of a particular dataset. And to simplify this, we could almost look at it as a kind of average of all the individual deviations within a dataset. Now, this definition is similar to some other calculations which look at deviations such as the standard deviation of a dataset. However, the variance is calculated by taking the average of squared deviations from the mean, which essentially means it is the average sum of the squared deviations. Now, because the variance value is the result of data being squared, the unit of measurement also becomes squared, which means two things. Firstly, the variance value tends to be larger than the data itself, and secondly, whilst useful for comparing two datasets using analysis of variance tests, commonly called ANOVA tests, it can't be used to visually represent deviation on the same graph as the actual data. For that, we need to use other statistical calculations which are covered in other videos. Now, before we move on, one final point to note. We should only complete a variance calculation on data that is normally distributed, where the data follows a bell-shaped distribution, with data values closer to the mean occurring more frequently than those that deviate further from the mean. Okay, so now that we know these key bits of information, we can start to look at how we actually calculate the variance, and we do this using a rather complex looking equation. However, once broken down into its constituent components, it becomes very manageable in terms of calculating the variance value. So what is actually happening? Well, the variance is essentially the sum of our squared deviations, so each data value minus the mean squared, which is then divided by one of two numbers. It can be the number of data values in our dataset, or it can be the number of data values minus 1. Now, how do we know when to use n, and how do we know when to use n minus 1? Well, it all comes down to whether the data values observed represent a sample of a larger population or the entire population, and determining this can be easier said than done. So a population in scientific terms refers to much more than a group of people. It is essentially an entire group that could be observed, whereas a sample is a subgroup of the population that could be observed. Now, obviously, it's better to look at an entire population because you are essentially analysing all of the data possible, but sometimes it's not possible because the population is too big, the experiment is too costly, or it is too time consuming to complete. For these situations, we use a sample of the population and use this to make inferences on the larger population itself. So as an example, let's say we take two 10 ml vials of blood from someone's arm, one before and one after an experimental treatment, and let's say we want to compare the number of monocytes in the two vials. Each vial is its own population, however it would be near impossible to count each and every monocyte in each of these populations, so we take a small sample from each and count the number of cells in these samples. Now, when it comes to our mathematical equation, this n minus 1 acts as a sort of correction factor, which is highly influential when there are only a few observations. However, as we add more data points to our dataset, it becomes less influential as we naturally get closer to the analysis of an entire population. So what do we mean by this? Well, if we were to make 5 observations which represent a population, then n would be equal to 5. But if these observations were a sample of a population, it would be 5 minus 1, meaning n would equal 4. If, on the other hand, we were to make 500 observations, which represents the entire population, then n would be equal to 500, and again, if these observations were a sample of a population, it would be 500 minus 1, meaning n would be equal to 499. Now hopefully from this you can see that if we divide a number by 5 and the same number by 4, we'd end up with quite different numbers. However, if we were to divide the same number by 500 and 499, then the difference would be relatively negligible, and this is what we mean when we say it becomes less influential the more data values you have. Now, to help make sense of our variance equation and what we've just learned, we are going to calculate the variance of 2 example datasets. So in our first example, we have a student who is counting the number of monocytes in a patient's blood sample using a microscope and hemocytometer. During their first count, they note 235 monocytes per microliter of blood. To try and increase the accuracy of their data, they perform repeat counts on different blood samples from the same patient, noting 204, 214, 256, and 251 monocytes per microliter. Following this experimental counting, the student calculated the mean to be 232, and now wants to know how dispersed their data is, i.e. what is the variance of monocytes per microliter. Now before we start transferring information into our equation, we need to determine what we are looking at and whether we need to use the equation for population variance or sample variance. So in this instance, the student is counting 5 different samples which come from a larger blood sample. Therefore, we are going to select the equation for sample variance, which we can now populate with all of the information from the question. So x minus x bar becomes our data value minus the mean data value, i.e. our deviation, repeated for each data value, and n becomes the number 5 as this is the number of data values observed. Now to make things easier, we can start simplifying this equation by calculating everything in the brackets on the top line of the equation, which still has some work to do. Our next step is to continue simplifying the top line of the equation by squaring each of the values in brackets, which just so happens to remove any negative values so that we are left with a number of positive values which can now be added together, giving us a value of 2054. Next, we simplify the bottom line of the equation, so 5 minus 1 is 4, allowing us to divide 2054 by 4, giving us a sample variance of 513.5, and as I talked about earlier, this value is larger than our actual data values. Now, interpretation of this number can be difficult, but in general terms, the lower the variance value, the less variation there is in the data. Basically, the data values are similar to the mean with little deviation. On the flip side, the higher the value, the more variation there is in the data. Basically, the data values are more spread out with larger deviations between the data and the mean. So let's have a look at a second example. Here we have a second student who is repeating the same experiment but with only a different patient's blood sample. The student was a bit slower in the lab and only managed to complete 4 cell counts, identifying 180, 217, 250 and 281 monocytes per microliter. Following this experimental counting, the student calculated the mean to be 232 and now wants to know how dispersed their data is, i.e. what is the variance of monocytes per microliter. Now, just like in our previous example, we are going to calculate the variance using the following equation, which can now be populated with all of the information from the question. So x minus x bar becomes our data values minus the mean data value, i.e. our deviation, repeated for each data value, and n becomes the number 4 as this is the number of data values observed. Now again, to make things easier, we can start simplifying this equation by calculating everything in the brackets on the top line of the equation. which can then be simplified by squaring each of our values in the brackets. And as we talked about earlier, the squaring of values removes any negative values from our equation. Now we can add up each of the numbers on the top line of the equation, giving us a value of 5,654. Next, we simplify the bottom line of the equation, so 4 minus 1 is 3, allowing us to divide 5,654 by 3, giving us a sample variance of 1,884.7. And as I talked about earlier, this value is much larger than our actual data values. Now, if we compare the variance of the data generated by student 2 in our second example to that by student 1 in our first example, we can see they differ from one another, with the variance value of student 2's data more than triple that of student 1, despite having the same calculated mean value. And if we bring up the raw data for each of these data sets, we can see that for the first example, the cell counts are closer to the mean, resulting in a lower variance value, which essentially means the data values in the dataset are less dispersed. Then on the flip side, the cell counts for the second student in our second example are further from the mean, resulting in a higher variance value, which essentially means the data values in the dataset are more dispersed. So what does all of this mean? Well, essentially the variance value allows us to see just how spread out and variable our data is. The more spread out the data, the larger the variance value. And with that, we come to the end of this basic laboratory calculations video. Hopefully you found this content useful, easy to understand, and can use it going forward during your data analysis. Thanks for watching, stay safe, and I hope you have a great day.